First of all, I want to thank the organizers for inviting me here today and for such a fascinating conference. And I've enjoyed it so far, and I look forward to the various other sessions that are going to happen today and tomorrow. So my name is Mohamady El-Gaby. I'm originally from Egypt, but I'm currently a researcher at the University of Oxford and a lecturer in neuroscience. And I'm interested in trying to understand how we learn, how our brain enables us to learn. And when we talk about learning, we're thinking about this wonderful ability to assimilate information from the outside world to help us change the way we perceive the world, but also change the way that we behave. And learning encompasses a wide variety of different types that range in complexity from the very simple to the very sophisticated and the very complex types of learning. And I just want to share with you an example of a very simple type of learning, that of classical conditioning. Someone said it at the front row there. Some of you might already be familiar with this experiment with Pavlov's dogs, essentially being hearing the sound of a bell every time they're given food. And so after some repetition, they begin to associate the sound of the bell with food, and you can measure this because you can see that they salivate every time they hear the bell. And it turns out that this kind of simple associative classical conditioning is ubiquitous across the animal kingdom. It's very common. Even very simple organisms like the sea slug can be taught in an associative way to learn very simple kinds of associations. And we know a little bit about how this happens mechanistically. But of course, learning can be a lot more sophisticated and a lot more rich. And I'll give as an example of this learning mathematics. Of course, we don't learn mathematics by learning just a group of associations. We learn mathematics by forming a knowledge structure that allows us to solve new problems that we've never seen before. And this is what I mean when I say flexible learning. And this involves not just storing information, but organizing this information into structures. And as a neuroscientist, I'm interested in what happens in the brain. Now, we know that the human brain contains billions of neurons with trillions of connections between them. And we know a little bit about how these neurons function. We know that they send electrical and chemical signals through which they communicate. And we know that one of the key sites of communication between neurons is the synapse, which is formed by one neuron onto another. And we think that this synapse here is key to understanding learning. We think that changes that happen at the level of a synapse might explain what happens during learning. And so the challenge that we are met as cognitive neuroscientists is to make the link between the brain and the neurons inside it and their operation and complex, flexible types of learning. And we want to answer the question of how do neurons in the brain mediate this flexible learning. And so I'm going to share with you today the approach that I've taken, and it's very much a multidisciplinary, multifaceted approach that combines a number of different fields. And some of the distinguished speakers this morning talked about the importance of interdisciplinary, cross-boundary work, and I fully agree with this, and I'll try and demonstrate an example of this. First of all, the most important aspect is to find an appropriate model organism to work with. Now, ultimately, we're interested in understanding human learning, human memory, but for various practical and ethical reasons, it's not possible to perform certain types of experiments on humans. It's not possible to have a certain level of experimental control over humans when it comes to trying to understand learning. So we need to find a model organism that's similar enough to allow us to address the particular question that we're interested in. And I choose to work with mice because they are simple mammals, so they have a very similar brain architecture to humans, and they have a very similar organization of their nervous system to humans, but they have the advantage of being simple to work with, and we have an incredibly powerful set of tools that allow us to not only record with very high precision from their brains, but also manipulate their neurons very precisely while they're behaving. The second aspect of this is to try and teach these mice to learn a flexible learning task. And so, of course, I'm not going to try and teach these mice algebra. I'm not going to, sit them in front of a laptop and try and to get them to solve calculus problems. But I need to think of something that gets at the heart of what a flexible learning task should be, but at the same time they can actually do. And so part of my research has involved designing and developing a task that mice can solve that gets at these cognitive abilities. And I won't go through much of the detail, but it essentially involves the mice responding to different cues with different behaviors within the same environment. Thirdly, it's important to be able to record the activity of neurons from appropriate brain regions as these mice perform these tasks. And so in order to do this, we implant microdrives that deliver electrodes into the brain region of interest. And we have to do this under a microscope, and it's quite a complex surgery that needs to be very precise because these animals are very small, their brains are very small. But what it does is allows us to record the activity of neurons from a mouse that is actively behaving. And what we see is these, what I've plotted here are some example data that we've obtained, where we've recorded from around 100 neurons, and this is just 2 seconds of recordings, and every time a particular neuron is active, you just see one dot. So we can very precisely find when the neuron is active and relate it to what the animal might be doing. But in addition to being able to record from these neurons, we need to also be able to precisely control these neurons. And specifically, we need to be able to control communication between the neurons. And so I've developed a tool that allows us to genetically modify neurons in the brain to allow us to control them with light. And it's a part of a family of tools called Optogenetics, but what I've worked on is to try and get these optogenetic tools to work at the level of a synapse, so at the level of the connection between neurons rather than the entire neuron. And what I'm showing you in this image here is a fluorescent image of 4 synapses. So this box, the length of this box is 1 micrometer, so 1/1,000,000th of a meter. And so we can target only those synapses, but not the neighboring synapses, using a way of genetically modifying them. And we can enable these synapses to be silenceable by light. So we can target them by shining a certain wavelength of light and switch them off when we want them to be inactive. So that's our overall approach. And so I'll speak a little bit about what we've actually found using this approach. And the first thing we looked at is to try and identify the neural signatures of learning. That is, what aspects of the activity of the brain actually correlate with the learning process itself. Now, I mentioned that we can record every time a neuron is active, so every time the neuron is firing an impulse. And one thing that we can do with this recording is relate it to where the animal is in space. So what you see at the top there is, in black, is the path of the animal in a particular box. And in red, each dot represents the activity of one particular neuron. So this neuron likes to be active around the middle of the environment. And we can quantify this, we can represent this in a different way at the bottom there with a heat map, where the red colors indicate a high activity, whereas the blue colors indicate a low level of activity. And what we find is, broadly speaking, 2 different types of representations. We see representations on the left there, which are representations of space. So these are groups of neurons that fire only when the animal is in a particular corner of the environment. And what we also see on the right are behavioral representations. So representations that are they have less information about where the animal is in space, but have more information about what the animal has to do behaviorally. And what we can do is we can follow the development of these representations and see if they somehow correlate with learning. And interestingly, we see that these behavioral representations in orange only emerge when the animal begins to actually learn, whereas the spatial representations are there before that, suggesting that perhaps these behavioral representations are a task-relevant pattern of activity. Furthermore, what's quite interesting is we can also see that the activity of these behavioral representations predicts performance. So if we look at the activity of these representations before the animal has to make a decision, we see that they're more active before a correct decision compared to an incorrect decision, as you can see in this plot here. So together, this suggests that we found a signature, to use the words of the previous speaker, a biomarker of learning. And so the next question is to try and understand how these biomarkers or these signatures form. And so in order to address this, we go back to our synapses. And one thing that we observe is that in adults, synapses differ in their ability to change, in their capacity for change. Some synapses are readily changeable, and others are quite stable. And we find that in mice, in the brain region that we're interested in, these different types of synapses have a very simple anatomical arrangement. So synapses coming from the left hemisphere are highly plastic, meaning they can change readily, whereas synapses coming from the right hemisphere are highly stable. Now, this is quite convenient because then it allows us to use some of our tools to go in and actually understand what a plastic synapse might be doing. And so I go back to my molecular genetic tool that I mentioned that allows me to silence specifically the synapses that I'm interested in. And we show that we can indeed do this in mice that are awake and are behaving. So you can see in green that the synapses that we want to silence have been silenced. The plot doesn't quite show that there's a shaded region in green, meaning when we turn on the light, these synapses go down. And when we turn off the light, the synapses go back to their original level. Whereas another group of synapses that we don't want to silence stay stable. And so we can very selectively target our synapses of interest. And what we see is when we silence these synapses, I'll skip this slide. When we silence these synapses, we remove these behavioral representations. And this was actually quantified in this plot. We see that representations have less information about behavior and more information about where the animal is in space. So we effectively remove selectively one type of representation from the mouse's brain. And at the same time, we also impair the mouse's ability to perform in this task. So what you see on the left here, when we silence plastic synapses, mice are severely impaired. They're effectively at chance. Whereas when we silence stable synapses, they're not impaired. Suggesting that these synapses that are highly plastic are necessary for the formation of these behavioral representations and for flexible behavior. And so, to summarize, we use a combination of recording and intervention combined with some cognitive behavioral neuroscience to understand the neural basis of flexible learning. And we find that changes in certain types of neural activity patterns correlate with flexible learning, and that when we remove communication via synapses that are able to change, we lose these activity patterns and we lose flexible learning. And I'm going to skip this one because it's essentially saying the same thing graphically. What I would like to say at the end is one of the initiatives that I'm quite excited about is the Muslim Researchers Network, which we've established in the past year, and I encourage anyone who is interested to join us, who wants to talk to other academics in various fields from across the globe to come speak to me. And another initiative, if you are interested in cognitive neuroscience, there is an annual meeting that happens in South Africa run by some of my colleagues from Oxford. And if you're interested, please go to this website and apply to that. And in the end, I'd like to thank my collaborators in Oxford and in Cambridge, and thank you all for listening.